So we are very happy to welcome the next speaker, Professor Arup K Indra, for the University of Portland in Oregon, USA. So Dr. Indra obtained his PhD from the University of Jadavpur in Calcutta in India in 2001. And then he moved to France, to the IGBMC Institute in Strasbourg as postdoctoral fellow and then as a senior scientist. He's now head director of the Department of Pharmacological Science at the University of Oregon State, and also professor in the Department of Molecular and Cell Biology at the University of California, Berkeley. His laboratory investigates the mechanism underlying crosstalk between skin cells and microenvironment, especially in atopic dermatitis, vitiligo, and skin cancer. So, Dr. Indra, very happy to give you the floor for your presentation. Thank you very much.
Yes. I'd like to thank all the organizers and Marik and also Elizabeth for helping me to really be here. And it's a great pleasure. And we can probably talk my little French in a little bit later. But I'm really honored to be here. And what I'm going to do is I'm trying to give you a quick overview of what we are doing, especially the concept about the connections between the lipids and the barrier establishment. So we all know, and we working between the barrier and cancer, what we call as the skin is kind of the junction between the beauty or the beast. So while we can have the tattoo on our hand, but we also have this most aggressive form of skin cancer, which is the melanoma, which probably we can discuss some other time. But skin is one of those organs which keeps our inside in and outside out. And so we all know that it has a multi-layered, the epidermal, the dermal, and the adipose layers. But then there are other innervations, the nerves, the sweat glands, so multiple cell types, and there are crosstalks between these different cell types in the skin. Now, we all know that it's also one of the ideal target for topical delivery of the drugs or the vaccines. Now, what is interesting though, the mechanisms underlying skin barrier, which keeps the inside in and the outside out, which is still, I would think, is kind of, we'll know a lot, but it's still a lot needs to be understood, especially in the concept of interactions between the lamellar bodies and the lamellar granules. We just heard from Professor Yamamoto. Now, in terms of the human epidermis, as you all, I would appreciate that there is a double barrier, a barrier that protects against from the external barrier, and there is an internal barrier. And so I'm going to shed lights about how we tried to, connected the dots and looked into the transcriptional regulations of these barrier functions which started way during my postdoctoral training with Pierre Chambon at EGBMC France. Now, one other thing that many of you would appreciate is that disease called atopic dermatitis. It's a chronic itch and it's an inflammatory skin disease, and it starts from early onset, and it's almost affect about 10% of the US populations, and also the world population significantly affected. And we all know that, but one interesting thing that I want to make here, that it is marked by increased proliferation and differentiation and impaired skin barrier function. But there is a dichotomy that there is a concept of inside out and outside in hypothesis that, so it's primarily a set of thoughts is that it's an inflammatory skin disease. What which is primarily initiated by Professor Peter Elias, that it's probably an outside in hypothesis, which is basically the disruptions of the barrier can trigger an inflammatory skin response. And I'm going to shed light to some of them as well. But what is also important to appreciate that patients suffering from atopic dermatitis develop called eventually over time asthma and allergic rhinitis, a process that we all know now is called an atopic march. And it's, of course, it's a big market for this kind of, developing products that can, mitigate and also help prevent the atopic dermatitis progression. Now, since my talk is primarily on lipids, so we all know that besides the skin proteins, there's the lipids and the bricks and the mortars, like as we have learned. So, so they are basically from an integrated structure, and so if there is a breach in this barrier, we often end up getting a lot of diseases. Needless to mention, some of these called lamellar ichthyosis, X-linked ichthyosis, and Harlequin ichthyosis. And there are multiple factors, multiple proteins and genetic transcription factors which are involved, either in directly or indirectly regulating them in an overlapping manner. And so this is a classical example of a patient suffering from a severe Harlequin ichthyosis, and then there is a moderate to severe Harlequin type 2 ichthyosis. But most important in this fact are the distributions of the lipids. So the lipids, which essentially, as you would appreciate, consist of primarily the ceramides, cholesterols, free fatty acids, and the triglycerides. And this goes way from the studies from Yoke-Bustra. And when you have the normal skin barrier, it controls the flow of the, so it helps in the retaining of the lipids and the liquids and the intracellular contents. But when there is a breach in this barrier and then altered the lipid distributions, the whole the barrier falls off. And so using the different, targeting different transcriptional regulators and epigenetic modifiers, we have shown that several of these is involved in regulating the epidermal barrier functions. And this is a macroscopic studies which shows that, if you remove the chromatin remodeling protein BRG1 or transcription factors such as TAF10 or TAF4, as shown on the right-hand side, this is an ex-gal diffusion assay. Many of you who have done studies during the development would appreciate that the mouse embryos on the right-hand side loses water. So in a dye diffusion assay, ex-gal diffusions assay, or the fluorescent dye diffusion assay, they are turned green, primarily indicating that they have impaired barrier. And then we moved on with these studies in my laboratory, and we found out that there's some other proteins which is called CTP1 or COOP-TF interacting protein 1, which are also the co-regulatory proteins which interact with some of these epigenetic regulators. We found that when you delete them, and I'm going to talk about two epigenetic regulators, CTP1 and CTP2. And then the assays on the right-hand side is basically telling you that transepidermal water loss. If you lose water, there is a significant loss of the water, and which is basically the hallmark of showing that they have impaired barrier functions. But more important point is to emphasize that they have abnormal epidermal junctional proteins, especially the corneodesmosomes, and we just learned, and I'm not going to go into the details. But more importantly, I like to emphasize that many of these proteins and enzymes that are involved in the sphingolipid biosynthesis, especially sphingolipid metabolism, like the ceramidase and ceramide synthase and fatty acid elongase, or ceramide glucosyl transferase, selective ablations in the genetic studies have shown that each one of them is responsible in altered barrier functions, increased transepidermal water loss, and also in some cases the lethal disruptions of the skin barrier functions and lethality, suggesting clearly that and emphasizing the role of lipids in this process of the establishment of the barrier function and their transcriptional control. Now, we know and we appreciate from earlier studies and from Yoke-Bustra's group that, clearly there exist the 12 different subclasses of ceramides, and which plays an extremely important role. And I'm not going to go into the details, but they are basically divided in terms of their fatty acid distributions, side chain lengths, and sphingosine contents, and the variable chain lengths. So you can classify them overall into 12 subgroups. And we can talk about that more in details later on. But we observed that using mass spectrometry and for lipidomic studies, we found that sphingolipid distributions in this mice, and I'm taking specific examples just to highlight the fact of the importance of the lipids. I'm not overlooking or just skipping things. That specific distributions of the lipids were altered in this mice skin, specifically when we looked into untargeted lipidomics. And that corresponded to specific genes that were altered at the transcriptional level when we looked into some of these lipid metabolizing genes. Some were up and some are down. And so what we did is we tried to integrate these studies in the transcriptional data, the lipidomics, and the transcriptomics data. And what we found that the three major pathways which are known to be involved, the de novo pathway, the salvage pathway, and the exogenous ceramide synthesis pathway, there were discrete enzymes that were altered, which were involved in this. It's a complex process. There are redundancy between different proteins, but there were key enzymes that we observed were altered, either up or down regulated, and we put it into more of a mechanistic approach. We found that there were, eventually it was leading to reduced sphingomyelin synthesis, reduced composition of the ceramides, and probably this was contributing to altered barrier functions, at least one of the contributing factors. Now, because of the time, and I would like to emphasize that there were other proteins. There are proteins which are involved in the gap junctional proteins, which I'm not going to discuss, indicating that there is just may not be the only functions that could lead to the altered barrier functions. But today's being more on the lipids, lipids definitely plays a critical role. And I'm showing these slides just to emphasize the fact that some of those lipid metabolizing enzymes were altered and regulated by CTIP1, but there were other proteins which was also altered and involved in this process. So similarly, we also found that CTIP2, which is a protein which is actually expressed in the stem cell layers, and there is a redundancy that in absence of CTIP1, there is an upregulation of the CTIP2. So it's a COOP-TF interacting protein, which is a transcriptional co-regulators. And we also found that when we selectively ablate CTIP2 in epidermal keratinocytes, we observed a similar effect. And when the CTIP2 mice, we looked into the distributions of the ceramides, we also found that there were distinct subsets of the genes that were altered, which was eventually correlated with the distinct distributions of the ceramides, the sphingomyelins, and in this individual de novo pathway, salvage pathway, and exogenous recycling pathway. And so, how much time we have? 10 minutes? Yeah, but then. Maybe not 10. Okay. Yeah. So one thing I would like to emphasize that in atopic dermatitis, there is always a growing concern about whether there is which comes first, the chicken and the egg. And so it's whether it's a barrier integrity which is compromised, or whether there is an inflammatory response that leads to the barrier disruptions of the barrier. But this has been nicely represented here. That there is not only, there could be either or, and it's hard to tease out. And I'm not going to go into that, but most importantly is that one of these important factors that is involved in this initiation of the process, which is the Th2 response, is triggered by the thymic stromal lymphopoietin. Now, when we knocked out CTIP2 in epidermal keratinocytes, and we found that this develops a spontaneous dermatitis, and that was triggered primarily through secretions of the TSLP. And it shows all the phenotypes that was observed in this CTIP2 epidermal, which is observed in adult skin. Now, as a method of the skin repair process, we can put emollients, or we can put, in the eczema, if you put just emollients, it may not be sufficient because you want to breach, and this is the breach in the barrier, you want to bridge the gap between the lipids and the external environment and the internal cells. And so, probably I'll just skip that. But what I want to emphasize that this is the vicious circle. And so it doesn't matter where it starts, but if you have an atopic dermatitis phenotype, either due to the breach in the barrier or increased susceptibility of Staph aureus, that can trigger an inflammatory response mediated by the T cells, which eventually connects through the B cells and immunoglobulin mediated response and recruitment of the mast and the Langerhans cells. So we used an approach which is called the untargeted lipidomics in human eczema patients. And we simplified the whole process by isolating lipids from single tape strips. And so on the lower panel, this is one of the representative examples that you could appreciate that in the eczema patients, there is a reduced intensity, which you can eventually correlate with specific lipids. And then in the normal individuals, the specific subsets of the ceramides or the sphingomyelins are altered or reduced. And when you translate that, we could clearly observe that in specific cohorts and subgroups, specific monounsaturated ceramides of shorter chain lengths were significantly altered. And this correlated also to their susceptibility of Staph aureus. What we observed that the sphingosine content was significantly altered without alterations of the sphingonine, and which is also one of the component of the lipid biosynthesis pathway. Now, when we correlated that with Staph aureus susceptibility, so we could have subgroups of patients which is AD Staph positive and AD Staph negative and non-atopic patients, we could find that there were specific subgroups of ceramides that were altered. And we can probably skip a lot of those. But most importantly, what we found that there were direct correlations of subgroups of lipids in human subjects, and therefore, based on the distributions of the lipids, you can actually correlate with specific subgroups which was probably remained unnoticed and unappreciated. And so, for example, we could see that specific alterations of cholesterol 3-sulfate in AD positive Staph aureus, but not in AD Staph negative patients. Compared to that, then excess cholesterol sulfate was found to be linked in the stratum corneum and X-linked ecthyosis. And so, as to put down the whole story short, we could found out that there were a subset of the free fatty acids that were altered, and this subset of the free fatty acids, some of them were also have a strong antimicrobial activity. And so I can show you a few things that, so what we came up with a process that if you can supplement some of these lipids, it can at least probably be effective in mitigating, at least from the context of the lipid content, some of the atopic dermatitis spectrum if you consider from the outside-in hypothesis. And so to test that, we developed a very simple formulations, and we treated them in a mouse models. And so, many of you might be aware of that if you use the oxazolone-mediated challenge, it can mimic in mouse models many of those processes that can be developed in human atopic dermatitis. So we did a head-to-head comparison with hydrocortisone and the two lipid compositions, which we call the LipoBell. And what you would appreciate here that when these mice developed eczema, this treatment with the hydrocortisone ameliorates most of these diseases, the redness, dryness, and eventually also at the molecular level, you see reduced immunoglobulin E. So does these two lipids. And so we since we are moving away more from the animal studies, so we did on some of the human volunteers. And this is just some of those example, is that, when you treat them on our subjects, and you could see that you could see a relief from the severity of the disease, at least in some subjects who volunteered, and then also in another examples. But because of that lack of time, I would like to summarize that, and we can talk later on during the break, but we have indications and we have shown that there are specific transcription factors and transcriptional regulators such as CTIP1 and CTIP2, which are key in controlling the epidermal barrier functions, primarily and at least in part mediated through regulations of the sphingolipid biosynthesis. And our data suggests that many of those could be direct transcriptional targets, could be indirect. Also, what we showed that altered lipid compositions, specifically some of the ceramides, cholesterol 3-sulfates, and specifically the free fatty acids, are altered in AD patients, which nicely correlates with Staph aureus colonization, Staph positive versus Staph negative. And it is probably tempting to consider that a lipid-based therapeutic approach could be used moving forward in the treatment of the skin defects and human diseases, especially in the case of the atopic dermatitis and ecthyosis. And I would like to end acknowledging all the people who are involved, especially some of the people in my group, Sean, who is a graduate student, and postdocs, of the collaborations with all the people at the dermatology, my funding agencies from NIH, the Department of Defense. And I'll be happy to take some questions. Thank you.
Thank you very much. Have any questions in the audience? Yes.
Thank you very much. I was wondering if you have any idea of how CTIP1 and 2 regulates lipid metabolism by keratinocyte.
Yes, so thank you. And so CTIP1 being a transcription factor, it actually regulates some of the subgroups of the lipid biosynthesis like sphingomyelinase, ceramidase, directly at the transcriptional level. And so as you see, when we integrated this through the RNA-seq and they did with ChIP-seq, it was recruited on the promoter of some of these target genes and regulate specifically in a negative way. So then in absence of CTIP1 or CTIP2, there was a functional redundancy. When you remove them, their expression was significantly downregulated in the skin. Now, we are looking into whether there are human patients where there is the loss of expressions or mutations in the CTIP1 or CTIP2. So we are in the process. We didn't see that there is like mutations, but we at least see that there is a loss of expression in patients where of CTIP1 and CTIP2 in atopic dermatitis patients.
Hello, thank you for your talk. So in your aim number 2, the last sentence mentioned, new lipid therapeutics proposed. What do you mean by this?
So I think what I meant was that if there is a possibility that we can, so mostly atopic dermatitis is being treated in terms of as an inflammatory skin disease. So IGF, so interleukin-4 and interleukin-13, these are the primarily one of the, the inflammatory cascades mediated through their receptors. So their antibody-mediated study is the neutralizations of this cascade can prevent. But what we think that this being the deficiency of the lipids could be detected at a very early process, especially in the children and in the young adults, by supplementations of the lipids that we do. So we all apply creams and the lotions, but probably they are not tailor-made for individuals who are susceptible, like those which have filaggrin mutations. I didn't show that. So filaggrin mutations is one of the important susceptibility locus in patients, besides a lot of other genes. So if by identifying which of the lipids that could be deficient in these patients, and by having a tailored lipid compositions, it's possible and it's feasible to create and prevent the initial loss of these barrier functions. And probably it is tempting to consider by restoration of this barrier, probably we would never get the subsequent IL-4 and IL-13 mediated response, which eventually triggers into a more chronic, like a whole body inflammatory cascades. So I didn't get a chance to really explain that.
I totally agree, but my question is more focused on which kind of chemical structure you propose. When you mentioned
lipid therapeutics, yes.
Which kind of lipids?
So it would be the essential. I didn't show that, but it would be essentially the free fatty acids. So we tried with the free fatty acids, long chains and the fat chains, free fatty acids, and essentially those are in the C14 through the C18 combinations. And so those are the ones, and we're kind of trying to find out which of those and what is the proportions by trying out different, variable, the ratios of these two lipids, but essentially the free fatty acids. Yeah.
If I may, question.
Yeah.
I've seen in your chips, I think it was chips
right.
for
right.
CTIP1,
right.
that a lot of targets are not in the promoter regions.
Yes.
Do you know if CTIP1 can act with cofactors to activate or modulate genes with a specific binding on, I don't know, intron or exon region?
Yes. Yeah. No, that's a very good question. So we haven't looked at those interacting proteins yet, actually, and we are looking at it. So the question was, are there other modifiers in interacting proteins in this, or in this complex, transcriptional complex, which interacts? We haven't looked at it, actually. But it's most likely there would be TAF4, TBP. That's our speculations, but we are looking at it. Thank you.
Okay, perhaps we have to move on. If you have any questions. So thank you very much.
Thank you.
Thank you very much, Dr. Indra.